Good morning, everyone, and welcome. I am delighted to have you here today. My name is Dr. S.K. Singh, and I will be guiding you through our presentation on calibration of UPLC system. This is an important topic in ensuring accurate and reliable analytical results. Today, we'll cover the key steps involved in calibrating a UPLC system, the best practices, and some troubleshooting tips. So, let's get started and explore how proper calibration can enhance our analytical performance. Let's talk about the importance of calibration in UPLC. Calibration is critical because it ensures our results are accurate, precise, and reliable. Without proper calibration, the data we generate could be misleading or inconsistent. It's especially essential for quality control testing, where every result needs to meet strict standards to ensure product safety and efficacy. Calibration also plays a vital role in regulatory submissions, helping us demonstrate that our methods are sound and compliant. Additionally, it's mandatory under current Good Manufacturing Practices, or CGMP, and pharmacopoeial requirements, which are the standards set to maintain quality across the industry. Lastly, calibration is a key part of the broader process called Analytical Instrument Qualification, or AIQ, which confirms our instruments are properly installed, working correctly, and suitable for their intended purpose. By maintaining rigorous calibration procedures, we uphold the integrity of our analytical processes and ensure consistent, trustworthy results. Let's take a closer look at calibration versus qualification. Qualification, which includes IQ, OQ, and PQ, is all about verifying that the system is suitable for its intended use. It's like giving the system a checkup to ensure everything is set up correctly and functions properly. On the other hand, calibration focuses specifically on measurement accuracy. It's about ensuring that the measurements we get from equipment like sensors or pumps are precise and reliable. For example, if we're looking at a pump's flow rate, calibration would be used to verify that it's measuring flow accurately. Meanwhile, qualification or performance qualification, or PQ, would confirm that the overall system setup meets the required standards for system suitability. Both calibration and qualification are essential. They work together to ensure compliance. Calibration keeps measurements accurate, and qualification verifies that the system as a whole is suitable for its purpose. So remember, both are required to meet regulatory standards and ensure quality in our processes. Now, let's talk about the calibration parameters in UPLC that are essential to ensure accurate and reliable results. First, we have the pump, which needs to be calibrated to maintain precise flow rate accuracy. This is crucial because the flow rate directly impacts the separation and detection of analytes. Next is the auto sampler, where we focus on injection volume precision. Consistent injection volumes help us achieve reproducible results across multiple runs. Moving on to the detector, calibration here ensures wavelength accuracy, which is vital for correct identification and quantification of compounds. Then, there's the column oven, which must be accurately calibrated for temperature control, since temperature influences retention times and peak shapes. Lastly, we can't forget about system pressure, which needs to be monitored and calibrated to maintain optimal operating conditions. Overall, thorough calibration of these parameters forms the foundation for high-quality, reproducible UPLC analysis. Let's now look at gradient accuracy calibration. This step is crucial to ensure that our solvent mixing is precise. We typically use a UV-absorbing tracer, like acetone, to measure the gradient profile. By doing this, we can verify that the gradient we're producing matches our intended profile. The acceptable variation here is within plus or minus 2%, which helps us maintain method robustness. If the gradient isn't accurate, it could affect our separation quality and reproducibility. Moving on to pump flow rate calibration, this is all about verifying that our mobile phase is being delivered accurately. The process involves collecting solvent over a set period and measuring its weight or volume. The key point is that our deviation should stay within plus or minus 1 to 2%, which is a typical GMP limit. This calibration is critical because accurate flow rates directly impact retention times and reproducibility of our results. Ensuring both the gradient accuracy and flow rate calibration are precise helps us maintain high method consistency and confidence in our data. Now, let's move on to detector calibration. First, we focus on wavelength accuracy, specifically for the UV or PDA detector. The method we use involves certified reference standards, such as a holmium oxide filter, to ensure precision. The key acceptance criterion here is a deviation of no more than plus or minus 1 nanometer. This is crucial because accurate wavelength detection ensures we are correctly identifying our analytes, which directly impacts the reliability of our results. Next, we have auto-sampler calibration. This involves checking the injection volume accuracy and the precision, which we measure using the %RSD, or relative standard deviation. Typically, we aim for a %RSD of 1% or less. Maintaining this level of precision is vital because it affects both the accuracy of our assays and their repeatability. Consistent injection volumes mean our data is dependable, and our results can be confidently reproduced over multiple runs. In summary, these calibration steps are fundamental for maintaining the integrity of our analytical method, ensuring that every measurement is accurate and reproducible, which ultimately supports the overall quality of our data. Let's move on to the next critical aspect, system pressure check. First, we need to verify that the pressure remains consistent and within the specified limits. This involves conducting leak testing to ensure there are no leaks that could affect the system's stability. Additionally, we should monitor for any pressure fluctuations, as sudden changes can indicate issues. When these parameters are within acceptable ranges, it indicates that the system integrity is solid. Next, we come to temperature calibration. It's essential to verify the temperature of the column oven regularly. We do this using a calibrated thermometer or probe to ensure accuracy. The acceptable variation here is within plus or minus 2 degrees Celsius. Why is this important? Because temperature directly impacts retention time and the shape of the peaks in our chromatogram, affecting the quality and reliability of our results. Now, for calibration, this process continues throughout the analysis. Regular checks and adjustments ensure our system remains precise. Remember, maintaining proper pressure and temperature calibration is fundamental to achieving consistent, accurate, and reliable analytical results. Let's talk about calibration frequency. Routine calibration is typically scheduled on a monthly, quarterly, or annual basis, depending on the specific requirements of the equipment and the criticality of the measurements. But it's important to remember that calibration isn't just a one-time event. After calibration, we need to consider ongoing actions like maintenance, repairs, or even relocation of equipment. The key point here is that the frequency of calibration and these follow-up activities should be determined based on a risk assessment, as outlined in ICH Q9. This means we evaluate the potential impact of calibration errors on product quality and process performance, and then set our calibration intervals accordingly. So, rather than sticking rigidly to fixed schedules, we tailor our calibration approach to the actual risk involved. This ensures we maintain confidence in our measurements, optimize resource use, and uphold compliance across the board. Let's talk about the documentation requirements that are essential for maintaining a robust calibration process. First, we have calibration protocols and reports. These are the detailed procedures and summaries that guide and record each calibration, ensuring consistency and traceability. Next, raw data records are crucial because they provide the actual measurements taken during calibration. These records serve as the factual basis for verifying calibration accuracy. Calibration certificates are also vital, especially those traceable to recognized standards, as they confirm that calibration has been performed correctly and meet regulatory expectations. Deviations and CAPA, or corrective and preventive actions, are important to document because they show how any issues are identified and addressed to prevent recurrence. Lastly, Calibration labels and status tags are simple but effective tools for quickly indicating the current calibration's status of equipment. All of these documentation elements are not just best practices, they are essential for regulatory audits. Proper documentation demonstrates compliance, ensures traceability, and ultimately supports the integrity of your calibration program. Remember, thorough documentation is key to maintaining trust and meeting regulatory standards. Let's take a look at some common deficiencies that often come up during inspections. First, missed calibration schedules are a big issue. When calibration isn't performed on time, it can compromise the accuracy of your measurements and data. Next, there's the lack of traceability to standards. This means that if your records can't clearly link measurements back to recognized standards, it raises questions about data integrity and reliability. Incomplete documentation is another frequent problem. When records are missing or not thorough enough, it becomes difficult to verify processes or results later on. Ignoring out-of-spec results is also a concern. Sometimes, deviations from expected values are overlooked or not properly investigated, which can hide potential issues. Finally, no trend analysis is a common deficiency. Without analyzing data over time, you miss the opportunity to detect patterns or early warning signs of problems. It's important to remember that these issues are often cited during FDA or EMA inspections. Addressing them proactively helps ensure compliance and maintains the integrity of your quality system. In conclusion, calibration plays a vital role in maintaining the integrity of UPLC data. It's not just a best practice, but a necessity to ensure that our results are accurate and reliable. Proper calibration helps us stay compliant with major regulatory standards like USB, ICH, FDA, and EU GMP, which is crucial for the quality and safety of our products. It's important to remember that calibration isn't a one-time task. It must be performed periodically to keep everything in check. And of course, all calibration activities should be thoroughly documented and easily traceable, so we have a clear record of compliance and quality assurance. By adhering to these principles, we support the generation of precise data, which ultimately underpins trustworthy analytical results. This, in turn, ensures that our products meet the highest standards and regulatory expectations. For further support, please don't hesitate to reach out to us. We're here to help you with any questions or additional resources you might need. And with that, I want to sincerely thank you all for your time and attention today. Your engagement is truly appreciated, and I hope you found this presentation valuable. Remember, our goal is to support you every step of the way as you apply what you've learned. So again, thank you, and please feel free to contact us whenever you need assistance.